The scientific studies of Daugavpils University Research Program Biology are implemented at the Institute of Life Sciences and Technologies and at the Center of Training and Research, Ilgas. The departments of Biotechnologies, Ecology, Applied Chemistry, and Technologies are situated at the Institute of Life Sciences and Technologies of Daugavpils University. Scientific laboratories of those departments are fitted with modern equipment and conform to laboratory safety guidelines and rules. The main areas of activity at the Genomics and Biotechnology Laboratory are plant nanobiotechnology, plant and animal genetics research, population genetics, gene expression, plant tissue cultures, and their application in breeding. The laboratory is equipped with a modern equipment that provides fast and high-quality analysis of genetic material, cultivation of plant cells and tissues, as well as mRNA research to improve the properties of important crops.
The laboratory is conducting research in the field of plant nanobiotechnology, which has a great potential for the practical applications in obtaining new genotypes for breeding purposes. We have all the necessary equipment to provide the entire technological process from sampling to the results of molecular analysis, as well as everything necessary to provide the use of plant tissue cultures for fundamental and applied research. The results of our research have been published in the top 25 of scientific journals, which allows us to be recognized on a European and global scale, as well as ensuring international cooperation. The technologies we develop have patents.
In 2018, the results of scientific research of the Laboratory of Genomics and Biotechnology are included in the 10 most significant achievements in Latvian science, and the staff of the laboratory has been awarded an acknowledgement by the President of the Latvian Academy of Sciences. Laboratory of Animal Ecology and Evolution studies the ability of living organisms to adapt to the changing environment. Research activities of laboratory staff relate to individual variations of behavioral and immunological reactions and population management. The laboratory has an infrastructure for keeping insects, together with devices for studying and storing samples. This includes climate chambers, fume hood, refrigerators and cryochamber, as well as precision instruments like microscopes and ultravox. EthoVision is the most widely applied video tracking software that tracks and analyzes the behavior, movement, and activity of any animal. It is designed to be at the core of the lab and offers integration of data and automation of behavioral experiments. 5 scientific laboratories of the Department of Ecology are located in the laboratory building of the Institute: Molecular Biology and Genetics, Aquaculture, Hydroecology, Zoo Culture and Nature Conservation, Parasitology, and Histology. Pond Aquaculture Laboratory Center of the department, located in Nagli, the largest pond aquaculture company in the Baltics, 120 kilometers from Daugavpils. Various molecular, biological, and genetic studies are performed in the laboratory. Cell stress, cell program death and cell memory, epigenetic changes in these fundamental processes, studies of genetic and epigenetic processes in wild animal and plant populations. Native, introduced, invasive. The modern research methods are used, including pyrosequencing and Raman spectroscopy. The Raman spectroscopy complex, Renishaw, which based on molecular vibration spectra and ready-made spectral libraries, allows the identification of individual molecules and the analysis of their changes. For example, we can analyze the changes in the DNA structure. Our flow cytometer not only analyzes cell changes based on fluorescence with the help of 3 lasers action, but also allows non-invasive sorting of cells. After then, these cells can grow as individual cell populations. DNA fragments, and RNA are isolated occurs with help of various centrifuges, including refrigerated centrifuges and ultracentrifuges. We can obtain DNA from various objects in a non-invasive way, from scales, from mucus, etc. In certain cases, a robot for DNA, RNA, protein extraction is used. DNA and RNA are analyzed occurs with the help of a variety of electrophoresis apparatus, including a capillary electrophoresis chip type, Agulen, and visualization by automated imaging system Biospectrum UVP block, and spectrophotometers Dnovix DS11 and Biospec Nano for analysis of 1 to 2 microliters DNA, RNA, DNA and protein solutions. We carry out very relevant research in the field of epigenetics. DNA methylation is a variety of congenital, physiological, and phenotypic changes that occur without changes in the primary structure of DNA as a result of various factors. Daugavpils University is the only university in Latvia with a laboratory fully equipped for automated recirculation aquaculture system research, with a total tank capacity of 40 cubic meters. For many years, the scientists of the laboratory have been researching the ecology of fish and crustacean species cultivated in Latvia in natural and artificial water bodies, recirculation systems, developing innovative technologies for adding value to aquaculture and production, using aquaculture byproducts, introducing new species in Latvian aquaculture, successfully implementing applied aquaculture projects. Researchers use portable multi-parameter meters provides extreme flexibility for measuring up to 17 different parameters. Designed specifically for the rigors of the aquaculture industry, the YSI Pro series instruments are true field meters that are simple, easy to use, and rugged. Digital smart sensors are automatically recognized by the instrument when connected. The Laboratory of Hydroecology performs functional and structural studies of freshwater ecosystems under the influence of various environmental factors using both field, instrumental, methods of physicochemical and acoustic analysis of water parameters, collection of hydrobiont samples, and laboratory, microscopy, chemical analysis of water parameters, molecular and biochemistry studies. Methods in close collaboration with the laboratories for aquaculture and molecular biology and genetics. The parasitology laboratory isolates, identifies, and studies various parasites, including zoonotic agents, using a variety of microscopic techniques, light microscopy, fluorescence microscopy, confocal laser microscopy, etc. The most commonly used methods are full parasitological examination, digestion and compression methods, various coprological methods. Confocal laser microscopy is used to visualize biological objects before being stained with various luminophores. In cooperation with Daugavpils University, chemists within the project, several protocols for visualization of parasite internal structures were developed. The results were published in Scientific Monograph. Light and fluorescence microscopy is used to study parasitic pathogens and identify species. Multifunctional automated microscope Nikon 90i is the most appropriate tool for such studies. The laboratories of the Department of Applied Chemistry have all possibilities for synthesis of novel, innovative emissive materials and for characterization and investigation of developed materials. We perform spectral investigations, obtaining for newly synthesized luminophores absorbance spectra and fluorescence spectra, steady state as well as time resolved and polarized, in various media. Then, in cooperation with biologists and biophysicists, we develop new methods for detection and research of living cells and species. As a result, methods for detection of amyloid fibrils and parasites are developed and patented. In laboratory of chromatography, intensive research on chemical analysis of environmental samples, fuels, food, etc. are realized. We have 8 gas and liquid chromatographs for solving various practical problems of chromatographic analysis, starting from control of environmental quality to developing of more efficient biofuels. The Technology Department has several electron scanning microscopes which are used in the studies of various biological objects and nanostructures, as well as nanostructures of biological origin. The Study and Research Center Ilgas hosts the Department of Biosystematics with 2 subdivisions, Center of Coleopterological Research and Center of Forest Biodiversity Research with its 3 laboratories. 3 scientific collections fitted with climate control systems are located in the Center of Coleopterological Research.
Here in Daugavpils University we have a huge beetle collection, and if it comes to tropical fauna, it's definitely biggest collection in the Baltic states. Also because of this fact, it allows me to create my doctoral dissertation here on site. I also should add that material for this collection annually is fulfilled with material from various expeditions where our colleagues are going every year. So also this collection is very unique. We have many species that is stored just here, and it's
basically amazing that we have here in Daugavpils such place.
The Center of Coleopterological Research provides seminar rooms and laboratories. Workplaces are equipped with microscopes, photographic and computer equipment, as well as software. For visiting researchers, accommodation facilities are provided. The X-ray tomograph is used in the field of biological studies where internal and external structures of biological samples are analyzed.
My current research revolves around using X-ray micro-CT system as a means of non-destructive imaging of 3D samples such as amber and bone. Our researchers are currently working on paleoentomological research, which is one of the most developed research fields in our university. They are collaborating with such institutes as Geological Paleontological Institute, University of Hamburg, Royal Saskatchewan Museum, Sneckenberg, German Institute of Entomology.
In the Center of Forest Biodiversity Research, there are specially equipped herbarium rooms for collections of plants, lichens, and mosses, as well as collections of snail shells and small rodent skulls. Researchers of this center have all the necessary software and equipment for field and laboratory studies.
Our study involves different spatial scales. We are studying forest epiphytes from the landscape level to molecular level, and this is very important to get all necessary technical support for the successful implementation of this project. And the Institute of Life Sciences at Daugavpils University is offering all necessary technical support for the successful implementation of this project.